morning, dear students. Today we are going to learn about transduction, another process for bacterial recombination. In the last class, we have learned about conjugation and transformation. They are also bacterial recombination processes. Then what is transduction? Transduction is a virus-mediated gene transfer in bacteria. Who discovered this transduction? Zinder and Lederberg in 1952. What is the basic process in transduction? From one bacteria, its genetic material or some genes is transferred to another bacteria with the help of a bacteriophage. What is bacteriophage? That we are going to see. There are certain viruses that is infecting bacteria. After infecting the bacteria, they multiply inside that bacterial cell, that is the host cell, and later led to the destruction or lysis of the host cell. Such type of bacteriophages are called virulent or otherwise called lytic phages, because it will destroy, it will result in the destruction of the host cell. And its DNA means the DNA of the bacteriophage. It remains outside the bacterial chromosome. And divide independently. Without depending the bacterial chromosome, it can have its own division. That is the features of virulent bacteriophage. Then one more type of bacteriophage also exists. That is temperate phage or non-virulent. Non-virulent means it is not destroying the host cell immediately after it infecting the host. But later, it also will destroy the host cell. First, it establishes a symbiotic relationship. Okay, and later only it destroy the host cell. Such type of phages are called non-virulent or temperate phages. And in the case of this temperate phages, in the time first it infects a bacterial host cell, the DNA of the bacteriophage integrated with the bacterial chromosome. At that stage means the bacteriophage DNA integrated with the bacterial chromosome. That stage is known as prophage stage. And it replicates along with the bacterial chromosome synchronously along with the bacterial chromosome. Okay, that's about non-virulent or otherwise called temperate phages. Then another term we need to familiarize, that is lysogenic bacterial cell. What are lysogenic bacterial cell? Bacterial cell that harbor or carry a prophage. What is prophage? I already told you. What is prophage? It is a stage in which the bacteriophage DNA get integrated with the bacterial chromosome. That stage is called prophage stage. So, a bacterial cell carrying a prophage is called a lysogenic bacterial cell. Okay, when the conditions become favorable, that integrated phage DNA leave the bacterial chromosome and they start to divide independently and later lead to the destruction of the host cell. Okay, and it is saying that this bacteria is immune to secondary infection by the same viral species. Then another two terms we need to familiarize, that is generalized transduction and specialized transduction. The transduction process mediated by virulent phages is called generalized transduction, and the transduction process mediated through temperate phages are called specialized transduction. We are going to see both these types separately. First, we are going to see generalized transduction. And all virulent phages are not carrying out that transduction. Then which all virulent phages carry out transduction? We need to understand some examples. Transducing phages. Example, E. coli phage P1, Salmonella phage P22, Bacillus subtilis phages PBS11 and SP10 are some common examples for the virulent phages that do transduction, general transduction. Okay, next we are going to see that process. This is a bacterial cell, and first it will get infected by a bacteriophage, and it injected its own DNA to this host cell, and after entering the host cell it undergoes multiplication and also lead to the breakage of the bacterial or the host chromosome. Okay, and after that, phage DNA get packed inside a protein coat to become new phages. And during that process, mistakenly, some segments of this host chromosome also get packed inside this protein coat. Okay, such phage particles that carries the host, that is the bacterial chromosome fragment. It is called a transducing particle, and this particle is doing the transduction process. You can see that the bacterial, that host cell is destroyed, and all these phages are released. And we need to take the case of only this transducing phage particle. It infect next host cell, and it injects its DNA into the host cell. After injected to the host cell, this fragment, we know that this fragment belongs to the DNA of the first host cell, get integrated to the second host cell. Okay. By that way, the DNA fragment from the first bacteria is get transferred to the second host cell with the help of a virulent phage. Okay. And one more point, why it is called generalized transduction, because it is not specific that a particular fragment of the first host bacterial chromosome get packed inside the protein coat. All fragments have the same chance to get packed inside the protein coat. So in that way, that process is called generalized transduction, because the chance for get packed in the protein coat for all the fragments is equal. Any portion of the bacterial chromosome can be transferred. So this type of transduction is called generalized transduction. Okay, I hope it is clear to you. Next we are going to see the specialized transduction. Specialized transduction, we already know that it is mediated by temperate phages. Temperate phages are those phages first is DNA. First it establish a symbiotic relationship with the host cell. When conditions become favorable, then it exits from the bacterial chromosome and enters a lytic cycle. Such type of phases are called temperate phases. It has two options. First option, we know that either remain integrated with the bacterial chromosome, and that stage is called the prophage, or it can stay separated from the bacterial chromosome to enter the lytic cycle. Two options are there for this temperate phases. Like coliphage lambda. What is coliphage lambda? Coliphage lambda is a bacteriophage that is infecting Escherichia coli. What is its feature? It occupies a fixed position in the bacterial chromosome. And that site, that position is called prophage site. Okay. It is a nature. Then we are going to see the process. This is the bacterial cell get infected with that bacteriophage. That is similar to polyphage lambda. After it get infected, the phage DNA enters into this bacteria and get integrated to the host chromosome. Host means that bacterial chromosome. Okay, that stage is called the prophase stage. We know that. And as it goes, got some favorable condition. For example, if that bacteria got some UV radiation, then what happened? That I will show you once more. Then this fragment exit from the bacterial chromosome, and that exit occurs with some error. Error means as it exit from the bacterial chromosome, it takes some portions of the bacterial chromosome also along with it. We can see that, and along with that the bacterial chromosome also fragmented. Okay, then it got packed inside the protein coat to form new phages, and it lies the host cell and release. And we are taking the case of one such newly formed phage. It is infecting another bacterial cell, and it is injecting its own DNA into this bacterial cell. And after injecting to this bacterial cell, this DNA fragment belongs to the first host cell, exit from that phage DNA and integrated to the second host bacterial cell. Okay, and only those gene that is located adjacent to the phage integration site only be transferred. That is the important thing. Because in the first bacteria, we have seen that the integrated phage DNA, as it separate from the bacterial chromosome, it takes some bacterial gene also along with that. So that is specific. Only those gene that is located near to the phage DNA only can be transferred to the second host. That is specific. So by that sense, that type of transaction is called a specialized transaction. Okay, clear? That is specialized transaction. Then we are moving to one more term, that is sexduction or F plus conjugation. What is F? Sorry, not F plus, F dash conjugation. This is an HFR cell. We know what is HFR cell. HFR cells are those bacterial cells in which the bacterial chromosome carry an integrated portion of F factor. Okay, and such a cell we are considering. And what is happening here? This F factor also exiting from this bacterial chromosome, like in the case of that specialized transaction. Here also, as it exits from the bacterial chromosome, it also takes off some portion of the bacterial chromosome. And that particle, that DNA is called a F dash factor. The F factor carrying some portion of bacterial genes are called a F dash factor. And if this bacteria, it mates with another F minus cell. F minus cell means it lack it lack a F factor. So it's called a F minus cell. Conjugation to be is forming, and this F dash factor is moving to this F minus cell. Okay, and we know that along with this F dash factor, some fragments of the HFR cell also present, and these genes don't need to get integrated to the F minus cell to undergo its replication. It can takes place its own replication independently. Okay, that type of transduction is called a sexduction. Okay, and in this process, this cell is partially deployed for the gene that is present in this fragment, and the same gene may be present in this bacterial chromosome also. With respect to that particular genes, this bacterial cell is partially diploid. Okay, this is sexduction. Sexduction is mediated through the transfer of F dash factor. That is the only difference, F dash factor. Okay, with this we finish this chapter and today's section. Thank you.
I'm an icon, man. And my girl about a 10. I should fuck her in the Benz.